good for the rest of you. Sorry for you. My apologies.
Got it?
Yes, you can go ahead and start. Go ahead and pass your simulator ones to the aisle, and as Professor Ahern is talking, I will just grab them on my way out.
Okay, guys, thank you for doing that. I'm happy to see you filling those out. Those are very important things, as I said, and you really can make a difference. And as Dan said, this is, something that if you get registered and you vote, people listen to you. And if you don't register and you don't vote, the same is true of student evaluations. Nothing's going to change. You've got to get out there. You've got to make these things happen, okay? All right. I was reminded at the very end of last lecture that I told you guys a lie. I said I didn't have one more thing, but I actually do have one more thing about hemoglobin. All right? So there is one more thing really cool, or maybe not so cool, but interesting about with respect to hemoglobin. And that is, and it's not even on my screen, but it's a very important consideration, and that's a genetic disease called sickle cell anemia. You probably all have heard much about. And I want to say just a little bit about that. So sickle cell anemia has another interesting Linus Pauling connection. Sickle cell anemia was the first disease that was correctly predicted to be a genetic disease. It was predicted by Linus Pauling back in the, I believe, 1940s. And he thought that what he saw this was that it was likely due to a genetic mutation, and in fact, he was correct. Okay, Linus Pauling was correct about an awful lot of things. Oh, by the way, today I'm going to be using this screen over here, and that's because I can't get this light turned off up here without turning out the whole darned room, and I'm got to figure that out. And this doesn't show up well on the camera, so I'm going to use this over here today. Anyway, he predicted that. He was right on in his prediction. All right. Now, sickle cell anemia arises from a very simple mutation. There's actually several different mutations that can give rise to sickle cell anemia. They arise as a result of mutation, usually, but not always, in the beta subunits. Okay? And these mutations give rise to one or two changes. One of the most common changes is converting a glutamic acid into a valine. Okay? So instead of having a glutamic acid at a particular position, a valine is present. And that very subtle change changes the behavior of hemoglobin itself. All right? What happens when that occurs is that hemoglobin no longer behaves the same way under low oxygen conditions. Under low oxygen conditions, individuals who have this mutation, their hemoglobin will polymerize, meaning that it will, instead of remaining as individual subunits, they will start making link. They're not covalent links, okay, but they will associate with each other and make a long polymer. The result of that long polymer is that it misshapens the blood cell that it's in. So blood cells, instead of being nice and fluffy and round and smooth, which is very important for them to pass easily through the capillaries. Remember, the capillaries are very tiny. Instead of having that happen, then those blood cells assume the shape of a sickle. They look like a sort of a quarter moon. They have sharp edges, and they actually get stuck in the capillaries. But what that means is under low oxygen conditions, like when you're running or you're exercising, you're using all that hemoglobin. The hemoglobin is giving up the oxygen. Hemoglobin starts to polymerize. The sickle cell shape forms. The blood gets blocked in the capillaries because the sickle cell gets stuck there. Well, that means that the tissues that need the oxygen the most, we spent all day yesterday talking about how the body was so well set up to deliver the oxygen to the tissues that need it the most. Now the tissues that need it the most can't get any oxygen. So what happens to individuals who have this, they can have, at the very least, very painful experience when they exercise, even moderately. Okay? And in some cases, extreme cases, this can result in death. Okay? So it's a very nasty mutation. It's called an anemia, interestingly, because anemia means lack of red blood cells. And so your body actually recognizes these sickle shapes as damaged red blood cells and takes them out of circulation. Doing you a great favor, right? Well, the great favor is now, as a result, you have fewer red blood cells that there are to carry oxygen. Now, an interesting story about about sickle cell anemia is that when epidemiologists started looking at where the primary places that sickle cell anemia were endemic, they discovered something interesting. And what they discovered was that the greatest prevalence of sickle cell anemia was in sub-Saharan Africa and tropical areas that were places where malaria were abundant. Okay? And subsequent to that, it has been shown that individuals who are heterozygous for sickle cell anemia, meaning one good copy, one bad copy, actually have a survival advantage when they're children. If you have full sickle cell anemia, meaning that you have both bad copies, it's not going to help you. In fact, it's going to shorten your lifespan. But in these areas of the world where you have a heterozygous individual, they have actually a greater survival through childhood. And it's protecting against that malarial parasite. The malarial parasite is an intracellular parasite that relies heavily on oxygen. And so this minor mutation, which would look on the surface to be very, very deleterious, actually has an advantage. Okay? It makes you think differently about disease because we think, oh, wow, that's unfortunate, et cetera, et cetera. But one of the questions people ask about disease, well, if this is so disadvantageous, how has it persisted in the population for many, many years? Why didn't it just get selected out? And it appears in the case of sickle cell anemia that there may be an advantage as a protection against malaria. If you're interested, it's in my head because last term I teach a class called Molecular Medicine. And one of the things we do in molecular medicine is the students in the class read a book called Survival of the Sickest. And it has all kinds of interesting stories like that about things that you might associate with, oh, wow, that's too bad, that's a genetic disease. And you learn that that genetic disease, in many cases, actually has an advantage. So it's a very interesting book. I can recommend it highly. It's called Survival of the Sickest. Okay, so that's my sickle cell anemia story. I want to finish the topic on 3D proteins by talking about protein folding. And I've been alluding to it, and I haven't really said anything about it. So let me say something about protein folding. I hope I've left you with the understanding that protein folding happens, okay, in globular proteins. It happens as a result of interactions between amino acids. They may be close, they may be far away, but this overall process relies on interactions between amino acids. Now, we know that protein folding is essential for protein function because I've already told you that the shape is essential for function, and the only way it gets its shape is if it folds properly. There are diseases that are known where the folding doesn't occur properly, and these are very deleterious. Okay? The most common disease that we hear of and think of where folding doesn't occur properly is called mad cow disease. You guys have all heard of mad cow disease. And that disease really puzzled people when they first studied it. They couldn't understand why they couldn't isolate a virus. They couldn't isolate a bacterium. They couldn't isolate anything that seemed to give this disease. But when they looked at the infected cells, they always found this same protein. And after scratching their heads for a long time, a person named Stanley Prusner, okay, said, it's the protein that's infectious. Well, there's nothing in biology that makes a protein infectious. How can a protein be infectious? Because to be infectious, it's got to get into a cell, but then it's got to duplicate itself. How does that happen? So it's a very big puzzle. People laughed at him when he first made this proposal. He called it a prion, P-R-I-O-N. He turned out to be right. It took people quite a while to figure out why he was right and how he was right. The first surprise came when they characterized the protein and they discovered that the protein was actually a normal cellular protein. It wasn't something that came from outer space. It wasn't something that came from somewhere else. But in fact, it was a normal protein that was commonly found in the brain of virtually every organism, including human beings. Human beings get a disease like mad cow. It's called Creutzfeld-Jacob. You don't need to know that, but that's what it's called. And it has very similar symptoms to mad cow. People go completely delirious. They die within a short time of getting this disease. They don't last very long. Okay? When they studied this protein, they say, well, this is a cellular protein. What's going on? Why is this cellular protein doing this? Well, when they looked in normal animals, they saw that they had the protein too. So what's the difference between the guy over here, the cow over here that's sick, and the cow over here that's completely loco? The difference was that the cow that had the disease had the protein folded improperly. And not only was it folded improperly, and here's the kick, one misfolded protein would interact with a properly folded protein and cause it to misfold. Then that second one that was misfolded interacted with a properly folded one and caused it to misfold. They form polymers not unlike we saw with hemoglobin in sickle cell anemia. Associate, 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 associate. When we look at the brains of mad cows or people who have Creutzfeld-Jacob, they have what we call amyloid plaques. Basically, the brain cells of these organisms have this protein just stacked, one on the other, on the other, on the other, on the other, and all of them misfolded. That's why it appeared to be infectious. Because once one got misfolded, it just propagated itself. And if it busted open a cell, then it just went and got into another cell and caused the problem. Now, there's a lot of concern about mad cow. Can you get mad cow disease by eating beef from a cow that had mad cow disease? Well, there's some circumstantial evidence that people argue about, but some circumstantial evidence that that might be possible. Yeah?
I read a study where they were feeding cows to fish, and the fish ended up eating them.
Yeah. So there's a lot, the circumstantial evidence, yeah, and you're right, but the circumstantial evidence for this actually is in human beings. In England, back in the late 1980s, they didn't inspect beef, and they had a mad cow epidemic. And a lot of mad cow beef made it into the English food supply. And about 5 years later, they had an increase in what they called unusual Creutzfeld-Jacob. It wasn't a standard Creutzfeld-Jacob, but it was unusual, and it was the same thing that was going on. People got very worried with that. Well, inspections are pretty important. Pretty important. When you hear people say we shouldn't regulate, I'll point to mad cow and I say, I want somebody looking at my cow to know if it had mad cow disease. And mad cow disease is really a problem because, yeah.
Sorry, I keep interrupting. So the problems, the symptoms, they don't show up till later.
It may take a while, yeah.
And with the way our food system is, we kill them before we know. So there's no way to really inspect them.
Well, so there's a big debate about this. I'm not going to go into that debate at this point. But yeah, there are a lot of considerations with this, okay? I'll say one thing that'll scare you, and then I'll turn the topic to something else, okay? This protein that misfolds, when it misfolds, is really weird, okay? It's really weird. How weird is it? Well, you saw ribonuclease yesterday? And ribonuclease, I said, had these really cool disulfide bonds that kind of gave it some structural integrity so that if I tried to heat it up, it still stayed stable. Mad cow disease really stays stable once that protein gets misfolded. How do you destroy the protein once it's gotten misfolded? Cooking will not do it. It will still stay misfolded. If you want to unfold mad cow misfolded protein, it takes over 700 degrees Fahrenheit. That's known as burning my food. Nobody cooks their food at 700 degrees Fahrenheit. Now, I won't say that you can get mad cow from your diet. There's people who debate it both ways, but it's worthwhile knowing at least what is out there. Okay. Protein folding is important. Protein folding is really interesting. Okay? Protein folding is important, and it's so important that cells have special complexes that allow them to help proteins to fold properly. You got some help out there. They're called chaperonins. How many of you ever went on a date with your parents, or you went on a date and you had to have a chaperone? Wasn't that the worst possible way you had to go out with a member of the opposite sex? Good God. Okay? The chaperone, in the case of a protein folding, provides a chamber in which a protein that's newly been made has a chance to fold without interacting with other stuff. It kind of keeps it from those distractions that it might get if it were interacting with other proteins. It's sort of an isolation chamber, and in isolation, that folding happens much more properly. Chaperonins are made in some very interesting circumstances, and they're made in virtually every organism on the face of the earth, which tells us that they are very, very important. Okay? They help proteins to fold properly. Where do we see them made? We see them made in cells that have experienced something called heat shock. Heat shock. What is heat shock? Well, E. coli, for example, really likes my gut. I eat a lot of food. E. coli likes the food. E. coli has adapted itself to my body temperature. It loves 37 degrees Fahrenheit. Centigrade, not Fahrenheit, both. 37 degrees centigrade, right? 98.6 Fahrenheit. E. coli is really happy. If I take E. coli and I raise it to the temperature of my body at fever, which would be maybe 40 degrees centigrade, okay, what I discover is that E. coli isn't very happy at that. It's gotten heat shocked. And it will start making chaperonins to help fold proteins properly. That turns out to be not trivial, because think about this. When I heat the temperature up just a little bit, what do you suppose is happening to some of those hydrogen bonds? They're getting broken. We're misfolding some things even with a slight temperature change. Kind of cool. Now there's a limit, okay? But for those slight temperature changes, chaperonins can come along and facilitate proper folding. Now, the last thing I'll say about folding, blah, blah, okay. The last thing I'll say about protein, I don't have good figures for folding, unfortunately. The last thing I'll say about folding is we would love to be able to predict folding. Well, we got the sequence of amino acids. We can get that from the DNA by using the genetic code. We know the sequence of every protein that's made in the human genome. Every single one of them. There's about 30,000. You do variants on them, there's about 150,000. There are about 30,000 unique proteins in our body. Okay? We know the sequence of them, and we can predict the exact structure of 0 of them. 0. We can predict where alpha helices will occur with reasonable accuracy. We can predict where beta strands will occur with reasonable accuracy. But we can predict the 3D configuration of 0, human, or any other protein. Why? Well, you guys already partly know the answer. What's a variable? How about turns? Turns happen in a variety of ways. So turns are going to be pretty critical. One turn, if I'm off on that one, then the next one's going to be off more, et cetera, et cetera. You say, oh, well, there's slight adjustments, et cetera. Well, question? Oh. When people first started working with computers, they said, oh, this is a trivial problem. We know the angles of all these things, and we can put it all together, and we'll get close to this structure, et cetera, et cetera. Until somebody sat down and said, well, how many angles are we talking about? If I take a simple protein, 50 amino acids, that's not very long, 50 amino acids, and I take into account all the possible angles of all the possible single bonds that could occur in there, and I say, give it to a computer and just randomly come up, because you can calculate the energy associated with each one of these, calculate what the structure would be on a random basis. They discovered something that kind of scared them. It's called Levinthal's paradox. Okay? Levinthal's paradox tells us how likely we are to be able to predict that structure of a protein using this random approach, going through iteratively through every possible angle. Okay? I'm going to ask you the question. If I took the most powerful computer on Earth and I gave it this problem for this 50 amino acid protein, and I said, go and solve the structure of this thing, how long do you think it would take it? 1 million years. Okay? 6 months. This is where you'll get astonished, okay? If you took all the computers on the face of the earth today, all of them working, okay, as fast as the fastest computer on earth today, we're talking about billions of computers, it would take 1 million times longer than the age of the universe to solve the problem. Nobody was right. Yeah?
So you saying, like, when we looked at hemoglobin yesterday, that was just a fanciful interpretation of what
No, that's the structure. I'm talking about predicting the structure.
Oh.
We're looking at actual versus predicted. Okay? So that tells us something really interesting. It tells us, first of all, that that folding process is really dynamic. And there's a lot of variables that we don't know about it. What we do know is something that you saw yesterday, which is that the sequence of amino acids is critical. And by golly, those things know how to put themselves into the right configuration, because they do 99.999% of the time, prions being an exception.
What is a prion?
A prion is a misfolded protein.
Okay.
Causes mad cow disease.
Okay.
Okay? All right. So folding's important. I hope I've communicated that to you. Folding's interesting, and there's a lot of people who would really like to be able to predict the structure of proteins, but they're not going to do it on an iterative basis because it's going to take too long. Okay. Let's turn our attention to protein purification. I added a bunch of page numbers from the book that I've got for you. One thing you're not going to find in the book is protein purification. So you're only going to have that here in what I'm going to be talking to you about. I think you'll find it's fairly straightforward. Okay? So far you've learned some... Oh, by the way, I'm going to finish early today because, again, I have to be somewhere, so you guys will like that probably. I know, it's sad. Yeah. So now I got everybody's attention, right? Yeah! I can pay attention. I only have to listen to him for another 5 or 10 minutes maybe, right? So, all right. So we've learned a lot about the properties of proteins based on the properties of those amino acids that were in the protein. We're now going to use that knowledge of those properties to allow us to separate proteins. Biochemists are really obsessed with separating proteins because what protein separation allows us to do is it allows us to get a protein away from all the other components of the cell and study that protein by itself. Divide and conquer. That's the sort of mantra of biochemists. All right? So we spend a lot of time figuring out the best way to separate proteins. All right. First one's pretty cool. It's called gel filtration. It's also called molecular exclusion chromatography. Gel filtration equals molecular exclusion chromatography. Right? It works by a very cool principle. Imagine I have a little tiny bead, about 1 millimeter across, little tiny bead. And that little tiny bead has some tunnels through it. Those tunnels are all exactly the same width. Okay? Exactly the same size opening, exactly the same width. All right? Now I have not one of those little beads, but I have thousands of those little beads, and they all have little tunnels, and all the little tunnels are exactly the same size. Okay? I take those little beads and I pour them into a column, like you see here, and I add some buffer, because I want my protein to stay in the right folded configuration, right? And I allow the buffer to flow through it, and what I discover is that proteins go through these beads in 3 different ways. Okay? It's the mama bear, papa bear, baby bear. Okay? The papa bear is a great big protein. All right? He's a humongo protein. He doesn't fit into any of the tunnels. All right? I'm stereotyping here, I suppose, but he doesn't fit into any of these tunnels. I can do this, I guess. All right? So what is his pathway through here? Well, his pathway is, I hit here, I go down here, I go down here, I go down here, I go down here, I'm out. Papa bear goes through there pretty quickly. Mama bear isn't nearly as chunky as Papa bear. She doesn't drink beer, she doesn't eat walrus and that sort of thing. Okay? You never seen a bear eating a walrus? Right? And she kind of watches her figure, right? So she fits into some of these tunnels, and they're kind of fun. And she's kind of keeping an eye on the baby bear, right? So she goes through some of the tunnels, not all of them. Consequently, she doesn't go through quite as fast as Papa bear. Baby bear, on the other hand, every tunnel is an adventure. Ooh, another tunnel! Ooh, another tunnel! Right? Baby bear hits every bloody tunnel that it can on the way down. Who's going to be the last one out? baby bear. Well, gel exclusion chromatography allows us to separate proteins on the basis of whether or not they fit into those tunnels. Those little tunnels have a specific size, and it's called an exclusion limit. The bigger the protein, the less likely it'll fit, the faster it goes through. This is the first one that comes through. There's Papa Bear. There's Mama Bear, or Baby Bear over here. Okay? The ones that fit in the tunnels go through very slowly. Okay, pretty straightforward. Let's imagine that I have another kind of, another bead, but now instead of having holes in this bead, I attach chemicals to it. Chemicals. Okay? The chemicals that I attach to the bead are up here. All right? I might have a chemical that I attach that has minus groups. There's all minus groups up here. It might be any one of these. And I might have thousands or millions of these on a given bead. The outside of that bead is going to look really, really negative. Or if I put these on there, the bead is going to look really, really positive. Okay? Now each of these, when they start out, has a counter ion. Right? So if this is negative, it has a positive counter ion, like maybe sodium. Right? This guy would have a negative counter ion, like maybe chloride or something. When I apply proteins to this mix at a given pH, what's going to happen? Well, those proteins are probably going to have a given charge, right? You've seen how the side groups have different charges. Let's imagine I've got a mixture of two proteins, one positively charged and one neutrally charged. Okay? I take that column and I take all those beads and I pour them in there, and I got a column that's, let's say, is full of negatively charged beads. If I had those two proteins, one that's negatively charged and one that is neutrally charged, which one is going to come through first? Negative. It's going to hightail its way through there because it wants nothing to do with that. Positive, or I'm sorry, the neutrally charged will follow a little bit behind it. It's not as much of a race to get through. What if I had positively charged ones in there? They're going to stick, right? So now I see I've got a way to separate molecule, or in this case proteins, on the basis of charge. Cation exchange resins will stick positively charged proteins. Anion exchange resins will stick negatively charged proteins. Yes, sir?
Will the sticking change the shape?
Will the sticking change the shape? Generally not, no. Okay? How do I get it off of there? Is it stuck forever? Well, proteins aren't making covalent bonds. So what's going to happen is it's going to come on, go off, come on, go off. It's going to travel through very slowly. It's eventually going to come off. One way I can wash it off is by adding a bunch of salt. Salt will start displacing it, start releasing the protein that's there. Okay, so this is called ion exchange chromatography, and ion exchange chromatography is a very powerful way to separate proteins on the basis of charge. I'm going to tell you about one more, and then we're going to quit for the day. This one's the easiest. Called affinity chromatography. Let's imagine I'm interested in a protein that binds to ATP. And I've got a mixture of a tube of all the proteins of my cells. Some of them bind ATP, some of them don't. I take a bead, and now instead of sticking something negatively charged on it or something positively charged on it, I stick onto it ATP. Well, when I pour that column, what's going to stick to it? Only those proteins that bind to ATP. I've all of a sudden separated the proteins that bind ATP from those that don't. Based on, and here's the name of the technology, their affinity for what's on that bead. This is called affinity chromatography. Things that bind to ATP are going to bind to that column. How will I get them off of the column? Wait a long time. If I want to get them off faster, what would I do? ATP. ATP is going to help get them off, right? Because when they come off and they grab ATP, now they just come floating off because nothing's holding them to the column. Make sense? Cool stuff, huh? Should we call it a day? All right. It can be done with anything. Anything that a protein binds to. Okay, see you guys on Friday.
Thanks.
You bet. Take care, guys.